Okay. Good afternoon, everyone. Today we will continue to talk about the first macromolecules in biochemistry. We have talked about the prokaryotic and the eukaryotic cell structure, right? So you know the difference between the prokaryotes and the eukaryotes. Okay. So today we talk about the first macromolecules in biology. You know, maybe in the breakfast or in your lunch, you have eaten fish, meat, or egg. But you know the major components for this food. What's the major component? What? What's the major component? Yes, very good. Okay. Even in your blood, here is a hemoglobin molecule. That means this molecule can carry the oxygen, right? So today we will talk about the amino acids and the proteins. In this section, we need to master the structure, characteristics, and the physical chemical properties of the amino acids. So in the first part, the first main point is for amino acids. You know, the proteins are made of amino acids, right? So the second goal for us is to understand the structural characteristics of proteins at different levels. You know what I mean, different levels? The protein structure can be divided into 4 different levels, right? So here we need to know the structural characteristics of the protein. And finally, we need to master the relationship between the structure and function of proteins. Usually we see the structure determines the function, right? So here is the main points in this section, amino acids and proteins. For the first part, amino acids. There are 3 keynotes here. 3 keynotes. The first one is amino acid. And Italian meals. You know what is Italian meals? Yeah, you know, for here is carbon atom, right? It can find with the carboxyl group and the amino group, right? And hydrogen atom and the side chain. So this alpha carbon atom are bonded 4 different groups. So to form enantiomers. You know, in humans, all proteins are made of amino acids, right? How many? Yes, 20 standard amino acids. So here you can see the keywords for the amino acids is the alpha carbon atom, and the R group is also the side chain. Okay. So besides, based on the different R groups, we can call the 20 standard amino acids like this. All of them are 20 amino acids. The first one is the glycerin, right? Glycerin is the basic, easiest amino acids in the 20 amino acids. And adenine, and valine, leucine, right? Leucine, isoleucine, and methionine, proline, cysteine, and phenylalanine. You know, phenylalanine is a combined word with alanine and phenyl. You know phenyl? Phenyl means what? Means a benzyl, right? You know, it's a specific group for the phenylalanine. And tyrosine. Tyrosine is another kind of amino acids. And tryptophan. Tryptophan is also an amino acid. Arginine, lysine, aspartate, glutamate and glutamine. It's different, right? And asparagine and serine and histidine. So here there are 20 standard amino acids here. Well, among these 20 amino acids, we can see especially in your typical animal cell, at a different level, you can see different molecules. Here is a cell level, right? The cell, membrane, and the subcellular organelles at the cell level. The second level is complex. In the center of the animal cell, here is nucleus. Nucleus consists of chromosome, right? Chromosome is nucleus. And for the cell membrane, the plasma membrane is made of the proteins, lipids, and carbohydrates, right? Especially for the proteins, protein level is a molecular level. It belongs to the macromolecules. That means a huge molecule, or big molecule, right? But you know, the proteins are made of amino acids. Here is the last level is monomolecules. It is the minimal molecules in the protein. So amino acid is protein. So we will talk about the amino acid. Now the amino acids, what is the definition for amino acids? What's the definition? Please. Amino acids are the building blocks of protein. That means the main what? Main bone, right? The main bone or the building blocks. That means the main structure of what? Of the amino acid. So amino acids are the building blocks of protein. It's the main structure. Based on this definition, we can divide the amino acid into the 20 standard amino acids. 20 standard. Just based on this figure, you can see in the center of these molecules, in the center is what? Alpha carbon atom, right? Alpha carbon atom is surrounded by four different groups, right? It's a covalent bond. The first important group is the carboxyl group. Another important group of amino acids. The amino acid is two words, right? The carboxyl group refers to a C, right? And the amino group means the amino. So amino acids includes two different groups: primary amino group and alpha carboxyl group. So here there are two different and two important groups for the amino acids. Another group is hydrogen atom. The last one is R group. We have talked about the R and also the side chain. Just based on this structure and the definition of the amino acid, we can form, or we can see the difference of the amino acid. The difference here is in enantiomers. You know what is enantiomers? In humans or in animals, we can only the L-type amino acid. You know, if we can see the L-type, you can imagine another type is what? The L amino acid. Another type is D. Yes, D. Most of them comes from the Greek word. The Greek word. L means what? Yes, level. And D, dextro. Level means yes, left. And dextro. Yes, right. It's different. I think so different. So, but in humans, proteins is only the L-type amino acid. But what is the difference between the L-type and the D-type?
Amino group.
What's the difference?
Amino group is in the left.
Yes. You can see from this figure here in the middle is a marrow, right? And the left indicates the L type iron, but the right one is a D type. You can use your hand, right? Just like this. Just like this, right? So here is the difference between L type and the D type. So because the alpha carbon atom is bound with four different groups. Here it comes to group, it's amino group, it's ethyl group, and hydrogen. So four different groups around it with the alpha carbon atom. So we call it enantiomers. Based on this structure, we can divide the 20 standard amino acids into four different groups. Four different groups. You can see here is the 20 standard amino acids is differ only in the structure of the side chain, right? Because they have the same amino group, the capsule group, right? So the difference is just between the R groups, the upper group. The first small group for the 20 standard amino acids is the hydrophobic anephilic amino acid. You know what is the hydrophobic? It is the amino acid is lacking of affinity for water, right? It's like you're afraid of water. So here is hydrophobic. And anephilic. Anephilic in chemistry refers to this molecule is made of the carbon and hydrogen, right? So you can see here, the highlight H is a hydrogen atom, right? It's hydrophobic, and here it's methyl group, So it's also hydrophobic and hydrogen. So you can see how many amino acids in this small group. How many? 1, 2, 3, 4, 5, 6, 7, 8. So there are 8 different amino acids belong to the hydrophobic aphelic amino acid. So this is the property of the side chain. That's based on the first small group of the amino acids. A second small group is also hydrophobic, but aromatic amino acids. Aromatic means what? Yes, you can see here. That's a benzyl group, right? A benzyl group, just like this. Here, the first amino acid is phenylalanine. The second one is tyrosine and tryptophan. So here is 3 amino acids. The 3 amino acids belong to the hydrophobic aromatic amino acid. In aromatic, there is another word we usually use, romantic. You know romantic? And so that means this molecule can what? This group can be used for fragrance. You know fragrance? Yes, many girl students or boy students use the fragrance. The fragrance because that means the molecule smells good. Smells good. Even for your own cell phone, for the iPhone, you know Apple company. And the iPhone, sometimes you smell this, it smells just like an apple. Okay, for the charger, if you charge it on the wall, sometimes the mouse will come to your charger and bite it. Do you have this experience? I got this experience in 2010. In my hometown, I charged my iPhone, but finally I can't see that my iPhone cannot be charged. And I found this line is broken because the mouse bite it. So this material is made of the aromatic material. It smells good, so many animals or humans can like it. So it's a hydrophobic aromatic amino acid. The 3 different amino acids here. Another type, another small group for this 20 amino acids is polar charged amino acid. You know what's polar? What's polar? Yeah, maybe you have learned in cell biology, the cell membrane is a polar, right? Because the membrane is made of the lipids. The lipids has what? Has a polar head and two polar tail, right? Has the lipids molecule. But for the amino acids, the polar, that means the R group, the side chain is what? It's polar. Polar. Charged. In the charged, there are two different forms for the charged amino acids. One is negative charged. Another one is negatively charged. Right? So here, there are three amino acids are positively charged. Positive charge is just, you can see here, lysine is positive charged, right? Because it has in the side chain, there are another amino group. Here, this amino group is a primary amino group, and here is the second, right? So for this one is another positive charge here, and for this. So here is a polar charged amino acid. For the negative, you can see here aspartate. Aspartate are these amino acids. On the side chain, the R group has another capsule group, right? Here is the alpha capsule group, but this one is another one. So this can be analyzed. So we can see this amino acid is negatively charged. That's like this. So here there are 3 plus 2 equals to 5 amino acids belong to the polar charged amino acids. The next part, or the next group is the polar uncharged. Uncharged, that means it has no charge, right? Has no charge. Has no charge. For here, you can see is hydroxyl group. Hydroxyl group is a polar group, but this molecule is not charged. It's not charged. Here is another one. It's opposite hydroxyl group. So here is there to see. So there are 4 amino acids belong to the polar and uncharged amino acids. For summary, these different groups, you can see here. For all the 20 standard amino acids, the hydrophobic, hydrophilic amino acids, there are 8 amino acids belong to this small group. And for the hydrophobic, aromatic amino acids, there are 3, right? 3 amino acids here. And for the polar uncharged, here, and the polar charged. So here it's totally different. You can just based on the side chain or third chain. So here is the amino acids in proteins. Here is the first part of the amino acids and protein, and you can see here. Another thing we needed to talk about, based on the protein, we needed to talk the based on the structure of the amino acids. You can see the capsule group belongs to the acid, and the amino group belongs to the basic, right? So we need to talk about the acids and basics, just based on the structure of the amino acids, bases, and the pH value. The buffers. In a buffer, what do you mean buffer? Just like here, the buffer is in biology, in biochemistry, but you can imagine the buffer in human biology. A human buffer means what? If you have a lot of fat, anybody or other people give you a strike, you feel no painful, right? Because you have buffer. The buffer is the strike. So, but in biology, especially in biochemistry, the buffer for amino acids refers to
Increase pH.
the what? Refers to the resistance to change of pH value, right? Maybe the peak value of the specific amino acid is 7. It can resist 6 to 8. So it's just near the peak value, right? So here's the buffer. Another one is ionization of amino acids. That means because it's two different groups, so each group can be ionized another. So here is ionization of amino acids. Okay, the keywords for acids and base. Acid-base ionization. That means this molecule for the amino acids is this group and this group can ionize. And the vector means is not only this group can be ionized, but also this group will also ionize. So here is two different. This one is acidic and this one is basic. So it's two different forms. So we can zeroize. And the titration curve. In chemistry experiments, you add some of the opposite group just to solution to add the different solution. Just here is a beaker. Is a solution here. The titration type you can add what? Add the basic solution or the acidic solution, right? So here the pH value in this beaker will be changing. So the titration curve and the buffer and the PI. The PI is an interesting word in biochemistry. Usually we use the PI refers to principal investigator, right? Especially in university or in your home country, the PI means the principal investigator. What means this word? It's just only in research field. But PI in biochemistry is a word isolate electric point. It's a point. So here is the key point for the basic and basis. You can see here is ionization of a typical amino acid because each of the amino acid has two different groups. This group, carboxyl group. Carboxyl group in bases or in acids, it belongs to the acid group, right? So this group belongs to basic group, right? The basic. So here, two different groups can be ionized. That's different. Here is the general structure for the amino acid. You can take your number for the first or the amino acid is here, is the glycerin. Glycerin is here. You can see different group. If only one of the carboxyl group ionized, just like this. This amino group is the same, right? The same. But here the carboxyl group ionized to what? We can ionize to the proton and what? The CCOH is carboxyl group ionized to proton and carboxyl group, right? So it's different. At this point, only this group can be ionized. So the parameter for this state, the parameter is pK alpha or pKa equal to 2.34. 2.34 is acidic pH value, right? Because only the carboxyl group ionized. So at this state, But in the opposite phase, you can see here is another group, amyl group allies, right? So the electronegative oxygen atom and the carboxyl group pull the electrons away from the amyl group and lowering the pK value. So here is a basic pH value. So the pKa equals to 9.6. So you can see these two different carboxyl group pK 2.34, and for the amyl group the pK 9.60. So totally, or in the middle here, the real state of this amyl of glycerin is just between 2 and 9, right? It's in the middle. In the middle. You can use another figure to illustrate this form. Here, the left one is no ionic form, no ionic. But the right one is a ionic form because this group and this group can be allied. And here to release a proton, and here to release hydroxyl ion. So two different. But just based on this, you can see because this 2.34 and 9.6, and if you add some other solutions, here is a figure. The x-axis indicates the hydroxyl ions, and the y-axis indicates the pH value. You can see from here 0, increase to the 9.60. So in the middle here, for this specific amino acid, here this amino acid is positive charged or negative charged. Negative or positive. Here, you can see here, okay, we can see the definition for PI. The pH at which amino acid has no net charge. No charge. That means the charge equals to 0, right? No charge here. It's no charge, right? This point, PI equals to 5.97. The term is isolated point. But you know, in the left of this PI, the left pH value equals usually pH less than the PI. The amino acid is negative charged or positive charged. Yes, very good. Positive charge. Because there are more protons in the solution, right? So it's positively charged. But if the pH more than the PI, the amino acid is negative or positive. Yes, not negative charge. So here is the PI definition for the different forms for this specific amino acids. So here you can see here, but just based on this equation, the PI 2.34. But you know the PI is not always the average of the small pK and the higher pK. Maybe you can calculate here 2.34 plus 9.60 equal to what? 11. It's almost 12, right? Plus combine together, almost 12. And here is an average of these two numbers, right? The two numbers. So the PI refers to a specific status of the amino acid. It has no net charge. That means the charge equals to 0. So here is the PI. Even for this figure, you can see here, for this PI value for left is acidic, but for right, shifted to right is basic, right? So here is the different. The pH value. But based on the property of amino acid, each of the amino acids has a specific PI. Here. But you can know, that each of them has a specific PI. You can see this PI can what? Can form a acidic and a basic pair. Because the amino acids have two different basic and acidic groups, right? So here is acid and a base conjugate pair. What's the function for this pair? This pair is able to resist the changes in pH of a solution. And that means if you put some specific amino acid in water, this water can form acidic base conjugate pair. This pair can resist the changes. How to understand the resistance the changes in pH value? You can see from this figure. You can see here is the pK equal to 2.8. It's just like the PI value, right? If the PI value equals to 4.8, it will resist the change in two different forms. One is increasement, another one is decrease, right? Increase here, that means more than 4.8, just like this. Maybe the 5.8, right? Another is decrease. Decrease that is less than 4.6. Maybe here is 3.8, like this. So here is the PI value, and here is the resistance range. You know the range? For example, this amino acid PI equals to 4.8. The buffer ability for this amino acids equals to what? 4.8 plus or minus 1. 4.8 plus or minus 1, that means the range is from 3.8 to 5.8. So this is the range 3.5 to 5.8. For the PI is 4. So that means the change minus 1 or plus 1 or plus 1. So that means the 4.8 is just between this number and this number, right? So here is the resistance. That means if you put this amino acid in a solution, the pH value of the solution equals to 3.8 or 5.8. The amino acid is stable. It's stable. So, but what is the function for the amino acids buffers in humans? If you got ill, maybe the body fluid, the pH value may change, right? Maybe change from the 6.2 to 7. There is a 0.2 changes, right? This change can be stable for most of the molecules in your body. So that means the amino acids created for the protein has a buffer ability. So you can see your textbook. The textbook, there is a table. There's a table. Please turn to page 36. The 36, there is a table, the pK values and the molecular weights of the 20 standard amino acids. That means the list all of the amino acids, the pK value. The pK value you can also from this slide. The first is the molecular weight, and the second is the pK value for alpha carboxyl group. Here is a specific parameter for the specific amino acid. And the pK value of the alpha amino group is two different. In some specific amino acids, there are side chain pK value. It is only a parameter, right? So you can imagine the pK value the pi, the isoelectric value just between the alpha capsule group pK value and the alpha amide group pK value. Here is the second one, arginine, the second one, the pK value for alpha capsule group is 1.82, but the pK value for alpha amide group is 8.99. 8.9. So the pi, the isoelectric value is just between 1 to 9. So here is the pK value of molecules in here. So here is the most molecules for the amino acid. The first part, the structure, the characteristics of the amino acids. And what's the physical chemical properties for the amino acids? It's not only a acid, but also a base. The base and the acid conjunct pair is able to resist the change of the pH value in solution. So that means the amino acids has a specific buffer. So here is the structures and the function of the amino acids. Okay, let's have a break, okay?